Hey everyone, quick biochemistry basics here. Let's talk about structure of protein. So the proteins have a primary, secondary, tertiary and a quaternary structure. Let's talk about each of them. Primary structure of protein. Primary structure of protein is a linear sequence of amino acids present in the protein. This linear sequence of amino acid is either written in a 3-letter or a 1-letter form. Now, these amino acids are attached with each other by a peptide bond. Now, the primary structure of protein is always written from the N-terminal of the first amino acid to the C-terminal of the last amino acid. The information of the primary structure of protein can be obtained by sequencing the gene that encodes the protein. This information can also be obtained by sequencing the protein itself. Secondary structure of protein. Now, in the primary structure of protein, the amino acids are linked by peptide bond, and the peptide bond has an oxygen attached with the carbon and a nitrogen attached with the hydrogen. Both oxygen and hydrogen have the ability to form hydrogen bond, and this results in folding of the peptide chain either in alpha helix or the beta sheet. Let's talk about alpha helix. Alpha helix is the most common secondary structure of protein. In this structure, the protein attains a spiral shape such that the side chain of amino acid remains outwards. The alpha helix is stabilized by hydrogen bonding. This hydrogen bonding is formed between the hydrogen of nitrogen and the oxygen attached with the carbon. Individual hydrogen bonding are of course weak, but collectively they are strong enough to stabilize the spiral structure. All the peptide bond except the first and the last participate in the hydrogen bonding. Each turn of the helix has 3.6 amino acids at a distance of 0.54 nanometers. Presence of amino acids like proline can make the helix unstable and disrupt the helix. The beta sheet. Beta sheet is the second type of secondary structure of protein. Now, in alpha helix, the hydrogen bonding was between the same peptide chain, while in beta sheet, the hydrogen bond is formed between the two different polypeptide or the same polypeptide chain. The beta sheet can be either arranged in a parallel or anti-parallel fashion. Tertiary structure of protein. Arrangement of protein in the three-dimensional space is the tertiary structure of protein. The tertiary structure will also have other bonds like disulfide bond, hydrophobic interactions, and ionic interactions. Quaternary structure of protein. The protein having tertiary structure can further interact with other tertiary structured proteins to either form dimers, trimers, tetramers, and so on. The structure so formed is called quaternary structure. Most of the proteins in the cell have the quaternary structure.